So up next, we have Ohana Cavalcanti Bezerra. Ohana, after studying biomedicine at the Federal University of Rio de Janeiro for her bachelor degree, received her master and a PhD in cellular and molecular biology from Oswaldo Cruz Foundation. Her dissertation focused on genetic epidemiology of infectious diseases, and currently she is a postdoc of CanSI Ontario Stage Host-Seek project from the Della Lana School of Public Health at the University of Toronto, studying genetics and epigenetics. The title of the presentation is Signatures of Natural Selection Affecting Disease Risk in Admixed Population. Take it away, Ohana. You're muted.
Yeah, now it works. Thank you for the introduction. My name is Ohana Bezerra, and thank you for the opportunity of sharing my work that was developed during my PhD in Oswaldo Cruz Foundation in collaboration with the University of São Paulo in Brazil. So pathogens, they have been co-evolving with human populations along of the history of human populations. And when we think on pathogens then that can be shaping the human genome, we usually think on the classical example of malaria. And I really like these maps here where we can see an overlap of the malaria endemic areas in Africa and the regions where there is a higher frequency of the HBS allele. The HBS allele is the allele that leads to the sickle cell disease. And the reason why the mutations for this condition is very frequent in Africa is because it confers a resistance to the infection of the parasite of malaria. So heterozygous individuals for the sickle cell or the sickle cell trait carriers, they have a survival advantage against malaria. And not only the mutation for the sickle cell, but other mutations in other genes, not only coding for structural genes of the erythrocytes, but also coding for enzymes, very important enzymes of erythrocytes. They also have this same pattern here. So they have a higher frequency in endemic areas of malaria. And today I'm going to highlight the PKLR, which is a gene that codes for the pyruvate kinase enzyme. That it's very important enzyme for producing ATP in the glycolysis inside the red blood cells. And the mutations of the PKLR, it impairs the glycolysis, as you can see here in this figure on the bottom. And because you have the glycolysis impaired, you're going to have a lower amount of ATP for the survival of the red blood cell. And in this case, these red blood cells, they are going to break. They are going to be destroyed in earlier days of the red blood cell lifespan, leading to anemia. And in this case, it is an advantage against malaria because the break of the red blood cells earlier in life are not going to permit that the parasite of malaria make their cycle of multiplication inside the red blood cell. So it confers resistance to the parasite Plasmodium, and it's a survival advantage in these malaria endemic areas. However, a couple of studies have shown that this destruction of red blood cells can increase the iron overload inside macrophages. Iron is a very important element of our body, and this is recycled by our cells. And because many infectious disease, the pathogens, they multiply, they have part of their cycle inside macrophage, iron overload could lead to the risk to disease that are provoked by intracellular pathogens. So here, the basic hypothesis that we have tested is mutations that confers resistance to malaria could be leading, on the other hand, to susceptibility to intracellular pathogens. And we have tested this in the model of mycobacterial disease. And I have used leprosy as the model of the complex traits here. Leprosy is a disease, is an infectious disease that it's very, that is endemic in Brazil, in Bangladesh, in India, and it has background involved with environmental conditions, and also a very strong genetics background. And leprosy is a very old disease that is still endemic in some populations. And this is a very tricky disease to study. After the infection of the mycobacterial leprae, the individual can go through different forms of the disease, mild form or severe form, and intermediate forms. And also, some individuals can still go to the reaction episodes of the disease. And all of these forms, they are regulated by the genetic makeup of an individual. And the idea of the works from the group that I made my PhD and I spent a great part of my life is to identify the panel of the genes that could trigger who can have higher chance to develop the severe or the mild form of the disease and make a better management of the patients. So in this study, we have performed a case control analysis. We have enrolled more than 5,000 individuals, and we have used the leprosy, case control with leprosy in the Brazilian population, and tuberculosis in Mozambique, an African population. We have accounted for population structure, and after performing the association analysis, we have also explored if we were able to detect signs of natural selection in the PKLR gene, and also performed some functional analysis. And starting with the SNP selection, here we have used the PCA as a start point of selecting our SNPs. And we have actually explored the PCA here, considering that we were inputting only the variants across the region of the PKLR, and knowing that the SNP weights, they show us how much each SNP contributes to this clusterization. We selected the top SNPs, the top SNP weights, and those SNP weights, they just gave us the SNPs with the greatest allele frequency difference between populations. And those clusters here that were formed in the PCA, we speculated that what should be in common here is that maybe those variants could have some functional relevance in the gene. And ranking for the top SNP weights, we could capture most of the intragenic diversity, and we have looked specifically for the PC1. We have selected SNPs where the genotype frequencies, they most vary between populations, which is good because we would like to go for the variants that we believed that some selection could be under selection in any of the population, specifically in African population. And after running the PCA, we have compared with the other steps here, allele frequency above 10%. We have considered pruning for high LED. We have annotated, and importantly, we have compared with the literature to see if the SNPs that we have selected have something already published considering malaria selection. And we were able to get some SNPs that was already published, and some SNPs that have previously suggestions of biological relevance in PKLR. So we ended up with 3 candidate genes. The first one is a synonymous variant in exon 11, and an intronic and an intergenic variant. So those 3 SNPs here, we have genotyped it in the population and performed our case control study. This is a case control study of gene candidate region, and I must say that we use the PCA here as an exploratory analysis, and this worked quite well for our study, but we are not sure if this method using the PCA can be good for discovering other variants for other genomic regions. And also, without using the PCA here, only using these next 4 steps, we have ended up with the same variants. So we believe that the PCA in our case added in our SNP selection criteria. Okay, so for the case control study, because we are working with the mixed population, the Brazilian population, we have to estimate the ancestry of these individuals. And so we have used a panel of 46 ancestry informative markers of the type indels in all the individuals of the study. And in our case control analysis, we can see in the right side of this slide here, we observed the association of the genotypes of those SNPs in the population of Rio de Janeiro. And after replicating in Salvador, we also observed that the synonymous variant is also associated with leprosy risk in Salvador. And red here is the significant associations. And on the contrary, we did not observe the association in Rondonópolis and Manaus, which are these other 2 populations of Brazil. And we performed a meta-analysis just to confirm if our results will go to the same direction. And indeed, we could confirm the meta-analysis using all the Brazilian populations that those SNPs are associated with risk to develop leprosy. And the last study here was the case control with tuberculosis. And we have a very small sample size here, so we were not able to identify a significant variation, significant association, but we could see the same pattern, the same direction of association. And then we have the concern that we have raised here, why we are looking for SNPs that are able to be detected associated with our phenotype of interest in Salvador, in Rio, but not in the other populations of Brazil. As I said, Brazilian population is the mixed population, and we have some specificities considering the different regions of Brazil that we are looking for. For example, south of Brazil, people here, they have a much more prominent European ancestry than people from the coast of Brazil, where Salvador and Rio de Janeiro, they were very important cities, port cities that received the most of the slaves during the colonization period. So these populations here, they have more African ancestry, while populations from more inside of Brazil, this African ancestry is diluted. So this model, considering that we are looking for a region that we believe that it's under selection in Africa, helped us explain why we saw the association in Salvador and Rio, but not in the other populations of Brazil. And looking for the haplotype analysis here, I highlight the TGG risk haplotype, where we can see it's very frequent in African populations compared to European populations. And we also observed that there is a higher frequency of this haplotype in cases of Rio and also in cases of Salvador. And we were able to get to see the association for the risk with significant p-values, while in Rondonópolis and Manaus, we were not able to see the association with the same pattern that we saw before for the individual SNPs. And in the tuberculosis analysis, we also see the same direction of association, and cases of tuberculosis, they have 64% of frequency of this haplotype, while controls, they have 54%. So with this, we consider that the PKLR is a potential gene that is associated with the risk of developing leprosy. And because we know that we have a higher divergence in the haplotype frequency between African and European populations, this was our first point to investigate natural selection in the region. So in order to find some genomic footprints of natural selection, I will highlight here this test, extended haplotype homozygosity test, where we have to consider that in neutral evolution, the allele frequencies, they are going to be randomly changed. But if you have an advantage allele that will increase in frequency very fast in a population because of survival advantage, then the nearby alleles are also going to have the frequency increased. And this is what we call hitchhike effect. So in this case, we see the selective sweep, which is an evidence of selection. So we tried to see that in using the 1,000 populations, Africans and Europeans. And we looked for an extended region, the flanking genes around the PKLR. And using the XP EHH, we know that values of this score above 2, they are indicative of selection. So we were able to find a region of almost 400 base pairs with 25 SNPs that presented selective sweep. And what was interesting here is that this evidence for selection, it favors the ancestral alleles in a gene that it's adjacent of the PKLR. And this selection was in Europeans and not in Africans. And interestingly, this region identified as positively selected in Europeans shows no evidence of the recently favored variant in Africa, where the alleles occur at intermediate frequency. So going further in the analysis, we saw that there are 6 SNPs here that are in strong allele with the PKLR SNPs that were associated with the mycobacterial disease. And moreover, these 6 SNPs, they are EQTLs for both of these genes in the GTEx. And just for curiosity, this HCN3 gene, it codes for a voltage-gated channel of ion transport, and is highly expressed in early erythroid cells. So here I'm just showing you the table where I have highlighted here in gray the 6 SNPs that presented the selection sweep, and showing that they are in high allele with the 3 SNPs associated with mycobacteria. And also highlighting here that they are QTLs based on GTEx, and we see that there is a decrease in the gene expression for the risk genotype carriers. And considering these results, we assumed, we hypothesized that individuals with the risk genotype could be producing less pyruvate kinase. So here just thinking on the risk haplotype, TGG people, they would produce less pyruvate, which would lead to a lower port of ATP for the red blood cells. And this is an explanation. Maybe this could be an explanation for the association that we have seen. And this is the summary of this hypothesis, and where we consider that the risk haplotype TGG, in this case here, we would have more red blood cells being broken, and the proteins that are regulating the iron homeostasis would be increased due to capture more hemoglobin and save more iron inside macrophages. And we have some evidence from previous studies from the lab that a higher amount of iron can be beneficial for the mycobacteria infection to growth. And in that time, we had in hand the serum measurements of iron and ferritin and aptoglobin. Ferritin is the protein that stores iron. And for iron, we didn't see any difference when we compared for the synonymous variant, looking for the genotype, the risk genotype. And we see some difference when we look for the ferritin in a group of health individuals, but these differences were able to be observed only in men and not in women. And for aptoglobin, we saw very smooth differences here in the way that we have expected. But all these analyses here, considering the iron measurements, we did not account for other covariates that can be modifying the level of irons in our body, as other genes or other covariates such as the diet and such as drug intakes and the female period. And with this, we believe that the best way to try to see these smooth alterations that we are defending in our hypothesis here would be looking for the intracellular environment. So we open some discussions for future works to confirm this. And the general idea that I'd like to bring with this study is how we can see in the present day disease, how can we explain, or how the present day incidence of some diseases, they can be a byproduct of the past adaptation to selective force. And these mechanisms we call the trade-off, where mutations for the resistance of one disease can lead to the susceptibility of other disease. And also using the mixed populations, we can increase diversity in genomics, and despite being very challenging to work with a mixed population, we can have we can have a good and rich resource for understanding how variants are affecting disease. And in our natural selection analysis, we give some directions, but we were not able to confirm that the PKLR region in Africans are subject to natural selection with the data sets and the tests that we applied in that time. But we were able to show that the PKLR gene can be a potential susceptibility gene for leprosy disease with a minor effect, as it has already been shown for many other genes in the disease. And here, if you are interested to know more about this work, this was published in 2021. So I would like to thank you for the attention, and also thank my supervisors, Dr. Milton Osorio Moraes, that has passed in the last year. He was a great mentor of this work, together with Antonio Guilherme Pacheco. Also, I would like to thank all my collaborators and friends from the Oswaldo Cruz Foundation that helped me so much and make my PhD a very nice period. and also the collaborations with the University of São Paulo, who helped in the evolutionary analysis. And thank you all.
Thank you, Hannah, for a great presentation. Let's see in terms of questions. Let's wait while we wait for a question in the Q&A. Have you had a chance to check some hypotheses around increased iron leading to increased infection in larger databases, larger genetic database such as UK Biobank?
No.
No.
Yeah, we didn't have the chance for looking for large database at that time. And I mean, I have right now this chance, but at that time I couldn't.
Okay. Do you still have access to the patient? And if so, could you consider doing a whole genome sequencing experiment?
Yeah. Actually, on that time, if we had a chance, I think one of the greatest approach for going directly to the point that we would like to show was to have more variants across the genome and maybe performing admixture mapping analysis. So yeah, I have access of those patients. We still have some other works that are coming soon, I hope. So yeah, but most of the studies at that time, we were looking for hypothesis-driven candidate genes.
Any questions for the audience? Have you so I will follow up with one more. So have you thought of the interplay between some of those variants and sickle cell disease?
You know, this is an interesting question because these days I have just saw a study where they were using both of the models to understand the mutations of the sickle cell disease. They were using the sickle cell disease together with the pyruvate kinase deficiency. Yeah, I mean, this is interesting, of course, but on that time, I didn't have information of the sickle cell disease in any of the data set that I could think of, but it's definitely something interesting to see if maybe some of these patients with the deficiency of PK, they have sickle cell trait. Yeah.
And whether it protects even more against malaria.
Yeah, yes. And we don't have much information on malaria studies confirming that, you know, the PKLR is associated with malaria. So this is something also that we need to address because we based our hypothesis on clinical papers, where people use it like a comparison of the variants and the haplotype of the variants of the PKLR in mild malaria, for example, and severe malaria, but you don't have like a whole genome or genome-wide association study confirming that the PKLR is associated with malaria. So yeah, we have in this work, I think we have many lines to explore more in future, and yeah.
Okay, great. So if there are no more questions, I would like to thank the speakers, Ohana and Warren. Thank you very much for such great presentations. Thank the participant for the seminar. And I would like to remind you that both talks are recorded and will be available on YouTube. And don't forget to tune in next month, June 6th, for two other fantastic presentations from Florence Chardon from the Shendur and Satira Lab at the University of Washington, and John Morris from Sanjana and labs at the New York Genome Center. So with that, I will say thank you to everybody for tuning in and see you next month.
Thank you.
Bye.
Thank you all. Bye.